Please welcome Moti Koren, Technion, Israel Institute of Technology.
Hi, everybody. Imagine a therapy that naturally finds its way to injured neurons, crosses the blood-brain barrier, and actually regenerates its spinal cord. That's no longer science fiction. That's NurExone Biologics. My name is Moti Koren from the Technion Business Unit with more than 20 years experience in the life science industry. NurExone is a Technion spin-off pioneering a new therapeutic pathway where conventional treatment failed. Its exotherapy platform regenerates neurons after spinal cord injuries, offering hope to millions of people around the world. The breakthrough was led by Professor Shulamit Levenberg, one of the Technion most distinguished scientists with a proven track record of turning academic innovation into real-world impact. So NurExone is not just a scientific breakthrough, it is the future of regenerative medicine and an example of an academic helping the real world. Thank you very much.
Breaking the wall of irreversible nerve damage. Lior Shaltiel, NurExone.
Thank you, everybody. Glad to be here, especially as a graduate from Munich. I have a PhD from LMU, as to get to be here back in Germany and be also live stream to my team in Israel and all over the world. I'm presenting today NurExone. I'm the CEO of the company, and we are using exosome-based therapy for the CNS damage injuries. How we do it? We are a publicly traded company, so we in Canada, so this is a disclaimer for the lawyers, but let's go to the science. So we have a beautiful, very beautiful body. It can heal itself when we deal with peripheral nerves. But what happens when we have a CNS, central nervous system damage? All knows that it's irreversible and we are just treating the symptoms, just the symptom. So it's very, very hard conditions like spinal cord injury, optic nerve damage, and brain trauma. And we are working on that field. What we're doing is actually targeting the P10 that inhibits this process. The costs are amazing comparing to this unique phenomena. We are talking about the family that's damaged, not only the patients. Around 1.2 million patients and families suffer from those CNS damage. We're talking between $1 to $5 million per patient, and the cost of our whole health system for CNS in the US is up to $7 billion. Let's go to the root, and we go to the quote of Christopher Reeve. The best way to restore function is to find ways to regenerate the nerves. And this is exactly what NurExone is doing. This is a MRI of the spinal cord, and we actually close the gap. We are regenerating the neurons in the area, and this model is a full transection model of spinal cord injury, and the nerves are regenerated in a minimal invasive way, in a short way, just 2 weeks after, 2 months, okay? And we can see functional recovery. Our aim is to get this to human next year after our very successful preclinical data. Those are the data that we see. 2 weeks to 1 month later, we can see on the right side a rat that can walk again. And we could do it in a full transection model of spinal cord injury. We could also use a compression model, which is just press the nerves, even with more and high efficacy. How we do it? We use exosomes. This is the tiny vesicles secreted from cells. In our case, it's the bone marrow mesenchymal stem cells. Those are tiny vesicles in a nanoscale, and we are loading them with the siRNA, short interference RNA, that inhibits specifically the P10 protein. So we are actually inhibit the inhibitor, causing proliferation of the nerves and regeneration. And this is our exotherapy platform. We actually doing 2 mechanisms of actions. We first switching on the regenerative activation of the cells by decreasing the P10 and using the exosomes as an immunomodulatory and a targeted missiles, so to say, into the injury because they are aiming to inflammation. We have a robust science. We have a very strong preclinical data. We have 5 families of patents, 2 of them already granted in the US, covering the administration route to the how to produce the exosomes, how to load the exosomes, and what to load into the exosomes. We have orphan drug designation in Europe and United States, and we are with a close collaboration with the agencies to get it to human. This is our portfolio. XOP10 is our lead compound, came out of the Technion with already 3 indications: acute spinal cord injury, optic nerve, and facial nerves, and we have other in our portfolio. The potential here is used, first of all, the drug, XOP10 for therapeutic. We use it exosomes only for a cosmetic or aesthetic longevity, and also the opportunity for licensing. We have a very strong team with many years of experience and a very strong scientific advisory board that comprised for neurosurgery to rehabilitation. And with that, the message is clear. We will by regeneration and create a functional recovery. Thank you very much.
Thank you. First question here. Detlef, please.
Thank you. Very interesting. What patients are you targeting? Acute spinal cord injury only, or are you also going for chronic?
We are going to the acute from 2 different reasons. First of all, it's true that spinal cord injury is the same name, but when you have a chronic patient, you have a glia, scar, it's completely different scenario. We are working on this 2 weeks of therapeutic windows that we have and try to reverse the damage and show functional recovery.
Why is yours working on mesoderms and all the others not?
It's first of all, it's not cell therapy, it's exosomes. And when you have exosomes, and it can be a different type of exosomes, each cells produce different. In our case, we use bone marrow mesenchymal stem cells, and we found it the most effective one. And they are targeting more specifically neurons, and this is why we see an effect.
Andrea is next, then we'll have Pavel and Andreas.
Question to the side effects. You mentioned that you inhibit PTEN, so this means mTOR is upregulated, and there is known, at least from in vitro, there is hyperproliferation, angiogenesis, and so on and so forth. So do you see any systemic side effects? And second question, can you target those exosomes? Because if you give them intranasally, they will go throughout the whole body. And if you have side effects, how do you target?
I will go with the second question, and then they are targeted by nature. Exosomes is a delivery system to communicate between cell to cells. They're going from healthy tissue to a damaged tissue. What is the target? Inflammation. The first things that they will do is reduce inflammation, and by reducing inflammation, they can prolong regeneration. Okay, this is the 2 things that we use exosomes. So they are very targeted. In our case, we have a proof of concept intranasally, but also intrathecally. So we can see that they accumulate in the area of the damage. To your first question, it's also related to the first question that I asked. We are using the acute phase. We are not giving it for chronic. It's very localized and very time-wise. So we are talking about 2 to 4 injections in the first 2 weeks, and that's it. It's maybe a wrong business model for business because we are only in the acute, but let's have this hope for those patients to get them functional recovery again.
Okay, we have a lot of 3 other questions. Please, Pavel, first.
I'll be very brief. Thank you. So just to understand correctly this short time window, does it mean that it's a hope for people who would have, the acute injury now, but people who had it 10 years ago, for example, there is no hope with your therapy? Did I understand it correctly?
That's correct.
Thank you.
That's correct.
Thank you, Andreas. Yes.
It's perhaps a bit unusual to see a public pre-IND company. So I'm curious now with your proceeds from your round that you're trying to pursue, what's your clinical development strategy now? I see that you are also orphan drug designated in the States and Europe.
Yes.
And what's the route through the clinical development?
So we are going therapeutic like in the FDA. It's part of BLA, you need to submit the IND. But to the question of being public, I think this is the question that needs to be in the room. We are thinking out of the box. We came to become public 3 years ago when we just spun out from the university. It's hard, I never heard something like that before, and we could raise, so to speak, close to $20 million like that. As a private company, I don't think that we will succeed in this ecosystem. So it is another proof of concept, not only the science, also the management, and also the way to raise funds in this very, very hard condition, especially to the hardcore biopharmaceutical. So the question is to go to therapeutic with a normal cell therapy approach, and also the cosmetic that can produce revenue during the time, like already next year.
Okay, and I'm sorry, Cyriac, we don't have the time for the last question.
000.
000. Great sounding. Thank you so much.